I'm Richard Casey, and we're filming at Colorado State University in Fort Collins, Colorado. And I actually have two roles at CSU. One is I manage the Cray supercomputer for the university, and I also manage the next generation sequencing core at CSU, where we do a lot of DNA sequencing. About 6 months ago, we brought in two new DNA sequencers from Life Technologies, the Ion Proton and the Ion Torrent. And basically, they have a chip like this that is basically a semiconductor chip on the back side. And on the front side, it has about 165 million wells for DNA sequencing. What we can do with this now is sequence almost human-scale DNA in about 15 hours and then run that over to the Cray for data analysis and processing. We had a project where they're looking at the Florida Everglades water sample, trying to identify endangered and invasive species in the water sample. And we ran some DNA samples on the next-gen sequencing chip, the semiconductor-based chip, and sent the results over to the Cray. We made about a 24-hour run on the Cray because it requires some high-performance computing to actually do the data analysis. And the output looks something like this, where they have a list of species in the water sample. And it includes things like bacteria and fungi and plants. The list we found was about 2,000 to 3,000 species long at several different sites in Florida. So integrating the next-gen sequencing with the Cray was really critical to make this project actually feasible and doable for this group.